We all carry this fundamental assumption about aging, don't we? It's
Hmm.
the slow, steady march of time. It's the continuous accumulation of wrinkles, the gradual shift from being able to run up 2 flights of stairs to maybe just one.
Right. We've always seen it as this time-dependent functional decline, a steady increase in mortality risk that you can just, map on a linear graph.
But that intuition, which has governed aging research for decades, it might be fundamentally flawed.
That's right. We have some fascinating new evidence, and it's rooted in detailed genetics and cellular biology. The studies primarily use model organisms like the fruit fly Drosophila and short-lived mice.
And they suggest aging isn't continuous at all.
Not at all. It appears to be a discontinuous two-phase process, and it's defined by a single, sharp physiological transition.
That is our mission for this deep dive. We're getting into the source material to answer the question, what defines that biological boundary? What is that tipping point where an organism moves from, let's say, healthy and coping to failing and, well, committed to the end?
And we're going to unpack three really interconnected insights that emerged from this research. First, we'll analyze the physiological marker that defines this transition, which has the strikingly memorable name, the Smurf phenotype.
The Smurf phenotype of.
Second, we'll look at the molecular level. And this is where we find something surprising, that age-related gene changes are actually a highly coordinated, well, a blueprint of poor condition, not regulatory chaos.
Don't find
And finally, we will explore the powerful promise, but also the serious peril of using epigenetic reprogramming to try and push that transition point backward.
The relevance here is so immediate, especially for you, our listeners, who want to stay well informed on this. I mean, understanding the precise moment an organism commits to decline offers completely new insights into how, and maybe more critically, when we should intervene with therapies.
It changes the entire target. It's not the whole lifespan anymore. It's one single critical window.
Okay, let's unpack this, starting with how that standard definition of aging was first challenged.
Well, historically, if you were studying aging, you'd track those continuous measures. So for instance, you could measure systemic inflammation markers from a young subject to an older one, and you'd see this steady, progressive increase.
Right. Or, in flies, I know researchers often use something called the negative geotaxis assay.
Which is a very technical term for a simple test.
It just measures how quickly they can climb a cylinder. Right. A proxy for neuromuscular function.
Exactly. And that climbing distance, it decreases continuously with age. That's the classic view.
So if aging is continuous, all your metrics should show that continuous deterioration. But the research that kind of broke this model, it used a clever, very visual trick to find that abrupt transition point.
The Smurf phenotype.
The Smurf phenotype.
It's brilliantly simple. It tracks intestinal permeability. So researchers fed the flies a non-toxic, non-absorbed blue food dye.
Okay.
If the fly is healthy, its intestinal barrier is intact, so the dye is restricted to the gut. The fly just remains its natural color.
But if that barrier fails,
the dye leaks into the hemolymph, which is sort of the fly's equivalent of blood, and the entire fly turns uniformly blue.
they look like the cartoon character.
Exactly, hence the name Smurf. It's this clear binary indicator of gut barrier failure.
So it's a physical, unambiguous marker for a failing physiological state. What did they learn when they started tracking this?
They learned that while the incidence of Smurf flies, naturally increases as the population ages, the individuals who become Smurf have something remarkable in common. Once a fly transitions into that Smurf state, call it phase 2, it is essentially committed to death.
Meaning what, exactly?
It enters a fixed remaining lifespan, regardless of how old it was chronologically when its gut failed.
Okay, let me stop you there because that is the most mind-bending finding in this whole study.
Yeah.
You're saying a fly that Smurfs at 20 days old has the same remaining lifespan, what's it, about 2.4 days,
About 2.4 days, yes.
as one that Smurfs at 60 days old.
Yeah.
So chronological time truly becomes irrelevant once that transition happens.
Precisely. The phase 1 state, the non-Smurf state, is where the individual's risk of transitioning increases with time, but their physiological markers are actually relatively stable.
So all that continuous decline we think we're measuring in the population average.
Is actually just the statistical result of more and more individuals making that abrupt transition. The individual non-Smurf, chronologically old fly, is physiologically nearly identical to a healthy young one.
And the moment they transition, all those previously continuous hallmarks like inflammation or motility, they suddenly show an abrupt change, not a gradual one.
They instantly look much, much older, and that's the key. This biphasic behavior isn't just some fly
It's
quirk.
been seen elsewhere.
Yes, the loss of intestinal barrier function, or its equivalent, has been validated across diverse species, from C. elegans to short-lived fish, and it scales to mammals.
The aha moment with that short-lived AKRG mouse strain really drove this home, didn't it?
It really did. So researchers were monitoring parameters like glycemia and rectal temperature in these mice. When they aligned the data to the date of birth, the parameters looked constant. There was no warning sign.
When they flipped it,
But when they realigned the data to the date of death, looking backward from the end, the data revealed this dramatic biphasic change. Every single mouse showed a profound shift within the last 40 days of its life.
and that shift coincided directly with the loss of intestinal integrity.
It proves that what matters isn't how long you've lived, but how far you are from this specific physiological endpoint.
Okay, so we have that when the Smurf transition defines the start of the end phase. Now we need the what. If the aging process has this sharp transition defined by a physiological state, what is happening at the molecular level? We usually assume that in old age, the genome just starts breaking down.
Right, that gene expression gets totally screwed up or dysregulated.
Yeah, random chaos.
And that assumption that aging is just a collapse into stochastic decay is what the transcriptomics data strongly challenges. The sources introduced something they call the plasticity hypothesis.
Okay, what's that?
It's the idea that these widespread age-related expression changes are actually a coordinated, purposeful, plastic response to a declining physiological condition.
So the body sees it's in a deteriorating state, a deteriorating soma,
Yeah.
and it responds with a defense mechanism.
Exactly. These changes might actually be mitigating damage, not causing it.
So the body is actively prioritizing survival. But if these gene changes are defenses, doesn't that make pharmacological intervention much more dangerous?
It does.
We might be knocking out the body's last line of defense if we try to force them back to a youthful state.
That realization is one of the most significant insights for anyone developing drugs in this space, and it's why understanding the source material is so crucial. They tested this distinction by running a really incredible experiment. They experimentally reduced the condition of young Drosophila females
How did they do that?
using large gene deletions that impaired fitness. And then they compared the gene expression patterns to those occurring in naturally aged flies.
And what they found must have been a massive surprise if the traditional model of random decay was true.
The concordance was huge. They found a substantial overlap of 414 genes and a highly significant positive correlation between genes affected by natural old age and those affected by experimentally reduced condition.
The conclusion was just unavoidable.
Unavoidable. The aging transcriptome is largely just a blueprint of poor condition.
What specifically was in that blueprint? I mean, what were the functional categories being changed in this coordinated way?
It's a textbook resource reallocation strategy. The genes being downregulated turned off were strongly enriched for energy-intensive processes.
Like what?
Think metabolic and mitochondrial functions like oxidative phosphorylation, energy generation, manufacturing amino acids, along with general cytoplasmic translation and making ribosomes, all the big energy spending stuff.
So the cell is saying, crisis mode initiated. We are pausing the expensive factories, cutting non-essential production, and conserving every joule of energy.
Precisely. And meanwhile, the upregulated genes, the ones being activated, were enriched for DNA damage response, immune activity, generalized stress response, and genome maintenance.
So the body is investing heavily in somatic maintenance and damage control when condition is poor. It's an organized, calculated defense.
Not molecular chaos.
And the evidence against just a random breakdown is very strong, isn't it? They found these changes weren't just random losses of function, but actual deprioritizations.
Absolutely. For instance, both aging and poor condition cause the downregulation of tissue-specific genes, like those active only in the head, and also female bias genes.
Which supports the idea of deprioritizing specialized tasks, like sexual or complex tissue traits, in favor of core generic survival.
And on top of that, these functionally enriched crucial genes were found to be tightly regulated. They exhibited lower expression variability and were subject to stronger purifying selection.
And just for clarity, purifying selection means these are evolutionarily critical genes.
Right.
The pressure of evolution ensures these systems are strictly maintained and not allowed to drift. It suggests the body intends these changes. They're not just random errors.
Exactly. The body is doing its best to adapt to a deteriorating state, which brings us right back to the source of that physiological decline.
Okay, let's trace this failure cascade back to the sharp transition we started with.
Yeah.
The decline of the intestinal barrier. The root of the Smurf phenotype. What's happening at the cellular level in the gut that kicks off this whole system collapse?
Well, the gut requires constant, massive renewal, and that's managed by intestinal stem cells, or ISCs. They are the engine of repair. But these ISCs maintain a very delicate and dangerous balance.
They have to be highly proliferative for regeneration,
Yeah.
but strictly controlled to prevent them from turning into cancer.
The inescapable cancer versus regeneration trade-off that, defines so much of longevity. As the fly ages, anti-cancer mechanisms are engaged, which unfortunately limits the stem cell's capacity to repair and regenerate. The ISCs accumulate cellular stressors, and the most profound finding is a metabolic switch. Old ISCs move away from efficient mitochondrial metabolism to glycolytic metabolism.
That's a huge shift. Glycolytic metabolism, relying on sugar breakdown outside the mitochondria. It's highly proliferative, but really inefficient. It's often called a pre-cancerous state, metabolically.
It is, and this also links directly to known longevity pathways. The TOR signaling pathway is necessary for stem cell division, but repeated activation inadvertently pushes these stem cells toward differentiation.
Which leads to eventual stem cell loss over time.
Yes, and this is why studies showed that reducing TOR activity, say, using a compound like rapamycin, helps maintain stem cell numbers in the intestine.
So the failure cascade starts with the ISCs getting stressed, shifting their metabolism, and eventually just running out of capacity to repair the tissue.
And the full epithelial breakdown follows a very clear progression. The gut loses its acidic barrier, which leads to metaplasia. This allows commensal dysbiosis,
Bad bacteria take over.
colonization by bad bacteria, yes, which drives inflammation, epithelial dysplasia, and ultimately the total loss of the intestinal barrier function.
Which leads directly to the Smurf phenotype. That's the single point of no return.
But here's where it gets really systemic.
Go on.
The sources show that the age-related inflammation of the intestine releases specific inflammatory cytokines UPD2 and UPD3. These cytokines travel through the body and impact the brain's antennal lobes, which are responsible for olfactory perception.
Wait, I always just assumed loss of smell in older individuals was purely a neural degeneration issue. Are you telling me that this basic failure of gut permeability could be responsible for a major systemic symptom of human aging?
It is a stunning connection. The failing gut is literally communicating its distress to the brain, contributing directly to the decline in the sense of smell we see in older individuals.
Wow. If the gut decline is communicating systemic failure, that suggests the whole system isn't irreparable, provided we can fix the underlying problem.
Yeah.
Which brings us naturally to the most exciting and maybe terrifying question. Can we reverse the transition? What about rejuvenation?
This is where partial epigenetic reprogramming enters the picture. This involves delivering transcription factors, most famously the Yamanaka factors, OSKM, or variants, to cells
To do what, exactly?
to reset the epigenetic clock, essentially turning them toward a younger, healthier state. The critical safety measure is making it partial reprogramming.
You want to avoid full pluripotency.
Exactly. You don't want the cell to reset all the way back to an embryonic state because that causes massive tumors or teratomas.
So how did they crack the safety code in a living animal?
They used a cyclic temporary expression protocol in mice. It was 2 days on, 5 days off, repeated over a period of 10 months.
A controlled, pulsed approach.
That was the key to safety. And the initial finding was huge. There were no adverse effects. And crucially, no increased incidence of tumors or teratomas compared to the control mice.
Okay, safety established. What about efficacy? Did the mice actually get younger?
They showed systemic rejuvenation across various tissues. They saw improvements at the level of the methylation clock, which is a leading biomarker of biological age, and it essentially ticked backward.
And functionally?
On a functional level, they observed improved repair processes in the skin, significantly faster wound closure in the treated mice compared to controls.
That's phenomenal. But the sources provide some really important, sobering cautions about the feasibility of applying this to humans.
Absolutely. The experts stress that reprogramming is most likely to be tested first in confined tissue contexts where gene therapy delivery can be tightly controlled.
Like glaucoma in the eye or heart fibrosis.
Exactly, where the therapy is localized. And there's also the threshold issue, which loops right back to our Smurf model.
You mean starting too late?
Yes. starting the treatment too late in life for the mice. This was starting at 22 months, which is quite advanced. It showed very limited molecular rejuvenation,
Suggesting there may be a physiological point of no return.
a point where the systemic decline is just too advanced for the epigenetic reset to be effective.
And this final caution brings us full circle, doesn't it? Right back to the plasticity hypothesis from earlier.
It does. Since we now know that many of the gene expression changes we see with aging are not regulatory breakdowns, but beneficial plastic responses.
Like that strategic downregulating of metabolism to conserve energy.
Right. So simply trying to force the expression of all genes back to a youthful state could actually be harmful.
Because if the underlying poor physiological condition is still there,
You're just reversing the body's coordinated defense system. You could inadvertently accelerate that Smurf transition. You have to fix the failing gut, not just mute the alarm bells.
so to synthesize this deep dive, aging seems to be fundamentally defined not by the passage of chronological time, but by a sharp, measurable transition into a phase of physiological decline, the Smurf state.
And that transition is marked by a sudden onset of symptoms and a highly coordinated transcriptional shift geared entirely towards somatic preservation and damage control.
The body is highly intelligent, even in failure.
It really is. And this leads to the idea that biological age is, well, it's fluid. Research has shown that markers of biological age can actually tick backwards when individuals recover from temporary severe stress.
Like major surgery or a serious infection.
Exactly. It suggests the physiological state is far more adaptable than we once thought.
Which forces us to ask the final, provocative question, linking everything we've discussed today, the discontinuous nature of decline and the coordinated nature of the response. If we could perfectly restore the physical health and condition of a 90-year-old to that of a 40-year-old fixing the gut, reversing the cellular stressors, would the molecular processes we call aging cease entirely?